Yeah. So, hello everybody. So my name is Corinne Abijas-Rizzo and I'm working in the Institute for Advanced Bioscience in Grenoble, in France, as a team leader, but also at the head of the department focusing on microelement, cell plasticity and cell signaling. And actually, last year, it was a big honor for me to attend this conference in Shanghai. And today, I'm very glad to chair this conference. I think the challenge today is to maintain this conference and to foster collaboration between China and France. And I think it's important to maintain this conference. I think it's a big challenge to do that in a virtual way, but I think we can succeed in that. So maybe now I'm going to present and to welcome the first speaker, Professor Xingyue Feng. So Professor Xingyue Feng spent a long time in US, and if I'm right, it's something like more than, it's 28 years. And it's a very long period in US through Maryland, San Francisco, and Houston. And then it joined the Life Science Institute in Zhejiang University in Hangzhou in China, and is focusing at elucidating the underlying mechanism and interplays among protein modification, signaling pathway, and gene transcription. And today he will talk about the mechanism of TGF-beta resistance in cancer. So, Dr. Feng, if you have, you can activate your camera right now and start your talk if you are ready for that.
I'm here. I need the host to start the camera.
Okay, so if you can share your screen too.
Let me try that screen. Yeah.
So you have something like, if I understood well, you have 15 minutes of talk, and then we will try to manage the question.
Can you see the screen?
Yeah, that's good.
Okay, great. Yeah, that's good. But I cannot turn my video on because you require the host help.
Oh, okay, so maybe something I can tell you, if something is wrong with, maybe I will be here with you, because if something is wrong, I will raise my hand. Okay.
Okay. Yeah, I can see you perfectly.
Okay. So
yeah,
thank you.
Hello everyone. Yeah, I'm Xingyue Feng from Zhejiang University. I thank the organizers for inviting me to share my research program here with my French colleagues and Chinese colleagues. So I have to say good morning to French friends and a good afternoon for my Chinese fellows. So I'm going to primarily talk about the TGF-beta signaling in cancer. Let's see, stuck. Okay, good. So TGF-beta signaling is looks very simple. With the ligand TGF-beta binds to two types of receptors on the cell surface and then activate downstream SMAD signaling. SMAD proteins are transcription factors as well to combine the DNA and turn on and turn off a large number of genes which are responsible for cellular physiology, such as cell cycle arrest. So this is the major tumor suppressor pathway. And this pathway need also be terminated during development and organogenesis. And during the last 20 years or so in my laboratory, we have worked on the receptor interaction, also identify the SMAD and also terminator, the phosphatase and nuclear exporter. And these are major players. At the same time, we also find that the modulators, the yellow one are positive regulators and the red ones are the negative regulators, which can fine tune the signaling strength and outcome from the ligand receptor to the SMAD to the gene expression. And the human genome actually encodes 33 TGF-beta like molecules. And there are 5 type 2, 8 type 1 receptors, and 8 different SMAD proteins. And in my talk, when I say TG-beta, I really mean the prototype TG-beta 1, TG-beta 2, and TG-beta 3. So you can imagine that anything that goes wrong in TG-beta signaling can lead to human disease. And we particularly focus on cancer in my laboratories. So the question is how TG-beta signaling is controlled in cancer cells. I mentioned that TG-beta is the tumor suppressor pathway. So the cancer, in order to proliferate, so they need to escape from TG-beta control. And this can be achieved in some cancers by simply somatic mutations in the receptor, in the SMAD2 or in the SMAD4. So you can imagine, and this will completely suppress the expression that in response to TG-beta, including the cell cycle inhibitors. And a perfect example is the mutation of SMAD4 in the pancreatic cancer and the colorectal cancer. So more than half of pancreatic cancer and about 1/3 of the colorectal cancer, the patients carry a mutation in the SMAD4 gene. The mutations are clustered in the MH1 domain, which is an important domain for transactivation, which is a transcription factor, right? And also another mutations may also impact the cofactor binding on the MH1 domain. So this explains a lot of activity, loss of activity of SMAD4 in pancreatic cancer and colon cancer. However, we have been able to, actually colleagues in the field as well, find that SMAD and TG-beta receptor genes are rarely deleted or mutated in a large number of cancers, except the GI tract cancers I just mentioned. So they include the breast, prostate, lung, liver, and leukemia, and lymphoma. So however, these cancer cells, they are resistant to TG-beta, either in vitro when in culture or in vivo. You don't see TG-beta target gene like cell cycle inhibitor expressed in the context. So there must be alternative mechanism that cause TG-beta resistance. So we in my laboratories, we found a number of oncogenes, and in the last few years, particularly 5 years, you found a number of protein tyrosine kinase. When they are already activated in the cancer cell, they can cause inhibition of TG-beta signaling. And one example I'm going to talk about is the LK today. So LK is either overexpressed or mutated or causing chromosome fusion. All of this will lead to the overactivation of ALK kinase. And this including such as the NPM ALK or EMF4 ALK in the lung cancer, this lymphoma, and here the point mutation is the neuroblastoma. This all leads to the overactivity, hyperactivity of ALK, which can help cancer cells proliferate in this cancer types. So US FDA approved a drug called crizotinib, and it's also second and third generation of ALK inhibitor, which is the ATP competing inhibitor. It occupy ATP site and inhibit ALK, and therefore can block the lung cancer cell proliferation. And in our hands, we found this ALK inhibitor indeed can slow down the cell proliferation. And accidentally, we found an ALK inhibitor can further sensitize the cancer cell to TG-beta. And here you see that the cancer cell do not respond to TG-beta well. TG-beta is supposed to suppress cell growth, cell proliferation. And with the ALK inhibitor, now TG-beta strongly completely block the cell proliferation in this lymphoma cell. We have tested a number of lymphoma cell lines, lung cancer cell lines, and the neuroblastoma cell lines. So you can see that the ALK inhibitors can enhance TG-beta signaling nearly in all the settings. Also test the gene expression of the cell cycle inhibitor here. So these are CDK inhibitors, including P15, P57, or P21. So they are not expressed in this ALK mutated or overly activated cancer cell line. When we add this inhibitor, this ALK inhibitor, all cell lines can express this TG-beta induced CDK inhibitors now. So they sensitize the cell to respond to TG-beta. And then biochemically, we found that ALK is a tyrosine kinase that can phosphorylate SMAD4 95 tyrosine, and this in vitro phosphorylation can be detected by the specific antibody we have made in our laboratories. And this is the in vitro. And you can also see that LK positive lines, you see the SMAD4 phosphorylation. The negative line, you don't see the phosphorylation. And in the positive lines, when we're locking down LK, you reduce SMAD4 phosphorylation, or you inhibit with the LK inhibitor, you completely inhibit SMAD4 phosphorylation. So this tells us that SMAD4 can be tyrosine phosphorylated in this LK positive cell lines. The outcome of this phosphorylation is the blocking of SMAD4's DNA binding activity. We have done a number of DNA binding experiments, including gel shift and in vitro DNA pull down assays. I'm not going to show you those data since our time is limited. I only show this chromatin IP experiment. In this, you see that the SMAD4, this shows the SMAD4 binding to the P15 promoter. So it's not induced by TG-beta. It's not induced by TG-beta on the P21 promoter or P57 promoter. Unless we're locking down ALK or we treat with ALK inhibitors like here, we achieve the same thing. So you deplete ALK, you can restore SMAD4 occupancy on this cell cycle inhibitor gene promoters. So that explains, I showed you early, that explains why the inhibitor or ALK depletion can enhance TG-beta signaling. So we have I will show you that the LK can phosphorylate the Y95 in the MH1 domain. So this is the DNA binding domain of SMAD4, and this phosphorylation is very close to the DNA binding hairpin loop. So this completely shut down the DNA binding of SMAD4 as a transcription factor. Well, we also analyzed the genome-wide transcriptional response to TG-beta. And here is a normal epithelial cells. We're locking out, it's crystal case line, we lock out SMAD4. You can see that the most majority of TG-beta responsive genes require SMAD4, right? And here shows that in this SMAD4 lockout cell, we put back the Y-type SMAD4. Now we can restore TG-beta response. Yeah, this is GLP control, it cannot restore. And the YE mutant, this is the mimic SMAD4 phosphorylation. You cannot rescue TG-beta response. And YF mutant can perfectly rescue TG-beta response. And on the right side, tells you the similar thing that tells us the SMAD4 phosphorylation inhibit TG-beta genome-wide transcription programs. And because of inhibition of gene transcription, so ALK can cancel the TG-beta induced cell cycle arrest. If we use the inhibitor, we can enhance gene expression, we enhance cell cycle arrest, and then therefore we can block the tumor genesis in the context of TG-beta signaling. And here, molecularly, we show that ALK can phosphorylate SMAD4 over here, and then this phosphorylate SMAD4 actually leave the chromatin. I don't have time to show you actual data. It takes along many cofactors, including P300 and CBP. So therefore, the genes will not be responsive to TG-beta with ALK mediated phosphorylation. And in mouse models, we'll also be able to recapitulate the tumor suppressor function of SMAD4. And here shows you the lymphoma induced cancer. We lock out SMAD4. We put a Y-type SMAD4 in there. And you still observe tumors because there's ALK in there, can phosphorylate SMAD4. If we put this mimic phosphorylation of SMAD4 in there, you cannot, you still observe tumor genesis, right? And if we put the YF mutant, which cannot be phosphorylated by ALK, now this SMAD4 mutant is a potent tumor suppressor. It completely shut down tumor genesis in this lymphoma models. And then molecularly, you see that the phosphorylation leads to loss of SMAD4 signaling, and the defective phosphorylation enhance TG-beta induced signalings, including induced apoptosis and suppressed cell proliferation, and also expression of cell cycle inhibitors, P15, P21, and P57. And this slide actually shows us what I have talked about in the last slides. I won't go over it again. And we also analyzed the tumor samples from human patients. We analyzed 41 human samples, and 27 of them LK positive and 14 of them LK negative. You see that LK positive, we see SMAD4 phosphorylation and only one outliers. And the LK negative ones, and the SMAD4 mostly negative. You probably were questioning why there's still 4 positives. So I can explain that if you are curious about this. So here you see that LK positive tumors, you see SMAD4 phosphorylation, and the tumor, the adjacent tissues are phosphorylation negative because LK is not there, is not activated, and even though the SMAD4 is equally expressed in all the tissues. Now here's our model in the normal cells, LK and TGF signaling, they are well balanced to control cell proliferation and the cell survival. And in the tumors, in LK positive tumors, LK is activated, and then they activate the canonical STAT3, MAP kinase, all the pathways, and they enhance proliferation and survival. At the same time, they shut down the tumor suppressor pathway, so T-beta SMAD4 can no longer suppress cell proliferation and tumor cell survival. I think my time's up. I don't want to have 2 last slides anyway.
Okay. Thank you very much, actually. Thank you. Okay, my video is off, so I cannot change anything. I think Milen.
Someone controlled my video. Yeah, I can.
Yeah. Okay. Great. Thank you very much for this very nice talk. And maybe I think you know the rule. So the speaker can raise the hand if they want to ask question, and the audience can address the question through the chat. And maybe I can start. I was very interested by your talk, actually, and I was thinking about, because you were talking about the TGF signaling, okay, and how the phosphorylation of SMAD can attenuate the suppressor activity of SMAD. And I was wondering, we know also the crosstalk between TGF and BMP signaling. So I was wondering whether when you did your genomic analysis, your transcriptomic analysis, if you were able to see any relationship with the BMP signaling.
Yeah, that's a good question. Yes, we actually analyzed BMP signalings in the context of SMAD4 Y-type knockout in the normal epithelial cells and the tumor cells. Loss of SMAD4 also cause inhibition of BMP signaling.
BMP7?
Yeah, BMP signaling. And we actually also did that in zebrafish embryogenic models. We showed the same thing. Loss of SMAD4 signaling by ALK. We overexpress ALK in the embryo. We can block both TGF-beta and the BMP signaling.
Okay. Okay. And do you have any consequence on ID1, for example, ID1 response and not this response? ID1
yeah,
is also.
Yeah, without SMAD4, ID1 is the expression of ID1 is largely reduced by loss of SMAD4. If we put the phosphorylation mimicking mutant in the cells, the ID1 can partially rescue, can still somewhere like 30%. So that tells us the ID1 is expressed in both SMAD4 dependent and SMAD4 independent pathways.
Okay. Okay. So Milen, do you have any question through the chat?
No question on the chat for now.
Okay. And from the speaker?
Yes, this is Pierre Renaud. Can I ask a question?
Yes.
Yeah, go ahead.
Yeah. Thank you very much for this really nice presentation. I read you had a beautiful paper about 1 year ago in I think in Nature about this. Huh? And the question I have is that the therapeutic effect of ALK inhibitors would be restricted to tumors that still retain SMAD4 activity?
Yes, this is actually a good question. I don't have time to talk about the actual outcome. So actually, in terms of lymphoma, inhibitor can restore TGF-beta signaling. That's pretty much sure. We also tried a lung cancer, which is more complicated. So as you know, that TGF-beta can suppress tumor cell proliferation in early phase, but it can actually promote tumor metastasis in later phase. So we did a model in the mouse lung cancer model. We found that this ALK inhibitor can enhance TGF-beta signaling and can actually help the spreading of the non-cancer cell. Okay, even though you can inhibit the proliferation of lung cancer cell. So this is very tricky. So we actually test our hypothesis right now whether we can use animal models or even in the future even human patients. In the case of ALK inhibitor, we probably need to add a TGF-beta inhibitor because you don't want to inhibit tumor growth at the same time you enhance tumor spread, right? So this is what we are trying to do in the animal model right now. Okay. And we're also trying to get a human specimen, which is hard because when people are treated with the ALK inhibitors, when they're recovered from the cancer, we cannot get their samples, right? Because they are okay. So we are trying to enrich the tumor cell, the circulating tumor cells from their blood, which we have a hard time to get enough for our experiments.